Hi, I'm Jonathan.
And hi, I'm Omar, and we're developing new technologies to edit life and cure genetic disease.
What if you could program a cell directly to fight cancer? What if you could solve the 7,000 different genetic diseases currently plaguing our species? What if you could increase the human lifespan to 100 years, to 150 to 200 years, and beyond with gene therapy and gene editing? Well, these types of science fiction ideas are now reality thanks to my guests today. Joining me on the Beat the Often Path podcast, we have Omar Abudayyeh and Abudayyeh-Gootenberg, creators of the AbuGoot lab at MIT. These folks are from little-known institutions of MIT and with their advanced degrees from a place you might have heard of called Harvard. So I'm just thrilled and deeply honored that they're joining an ignoramus such as myself today to share what is truly cutting-edge, groundbreaking technology with incredible implications for defeating cancer and a wide range of other diseases. It just doesn't get more cool than this. So I hope you will tune in to this episode, and thank you for joining me on the Beat the Often Path podcast. All right, well, welcome to the show, guys. Now, I consider myself a pretty smart person because when I microwave frozen foods, I'm able to set it to the 50% power setting to better de-thaw my food. So, I've looked at your bios and, we were talking about MIT, Harvard, various fellows, all of these things. So, I would like to say that we're intellectual equals here, but I know that that is absolutely not the case with me being clearly the smarter of the group. But can you explain to me a little bit about what it is that you two have been up to and why it's so relevant?
Yeah. So, right now, medicine is kind of going through a lot of changes with new technologies. And in the past, many people who are born with genetic disease could have treatments, but it was impossible to actually go into their genomes and fix that letter or problem in their genome that led to that disease. There were no cures available. And now we're seeing technologies that are truly programmable, that allow us to program those technologies to go into the genome, which is really 3 billion letters in all of your cells, and be able to actually change any errors that lead to this specific disease and then actually cure it. And so it's really, that's what we're starting to see now. And the technology that's gotten the most coverage that does that, called CRISPR, this new genome editing technology, is really the first wave of a whole slew of different programmable medicines. So in our lab at MIT, we work together to develop new programmable medicines that we hope someday will be therapies for genetic disease, for other common diseases like cancer or cardiovascular disease, and enable a new wave of treatments and medicines that can really help alleviate a lot of the suffering that people have from many of the health problems that come from these causes.
Okay, so, CRISPR, for many people, myself included, CRISPR is what you think of with a vegetable crisper in the refrigerator, something that you don't know how to use, but you realize that you probably should. What is CRISPR and why is that so significant?
So, CRISPR actually describes a naturally occurring system that originates from bacteria and archaea and, basically other single-celled organisms. And so it's interesting because it took a really long time to understand what CRISPR was. I think as we started sequencing more and more life in the 90s, starting with the genome, and people just started sequencing all sorts of bacteria and environmental samples, we started collecting this repository of genome information. And one thing people quickly noticed, I think the first observation was actually in the late 80s, and people started seeing it more in the 90s, was that bacteria had these really weird repetitive sequences in their genomes. And no one really knew what they were, but it took the sequencing revolution of the late 90s and early 2000s to basically start noticing that the repeat sequences, in fact, the sequences between the repeats that were in bacteria were starting to show up in the viruses that infect bacteria. And people thought that was really curious that there was this connection between bacteria and viruses. And I think in one paper, someone even remarked, well, maybe this could be some sort of immune system in bacteria. And we're seeing marks of previous infections scarred into the bacterial genomes. And lo and behold, that sort of early prediction turned out to actually be true. People showed that when bacteria were infected, the surviving ones could store pieces of their infections in their genomes, and those pieces would actually be expressed to then fight against future infections. And they actually were used to cut up the viruses if they ever showed their face again, basically. So
Like
it
the
was kind
signs
of
in
an
a
adaptive immune
7-Eleven that
system.
said, "Don't let this person in the store."
Exactly. And so like humans have a version of this, what's called adaptive immune systems. So we have antibodies, right? And when we get infected, the hope is we have some sort of antibody recognition. If we do, then we can overcome the infection, and then we store those antibodies for future use, right? That's literally what CRISPR is, but just a different mechanism. Instead of proteins, like antibodies recognizing proteins of viruses, it's a system to actually recognize the DNA or RNA of a virus and cut it up. And the really interesting insight was, there was Jennifer Doudna and Feng Zhang, where we did our PhDs with him. Basically around 2012, they both published papers showing that they realized that this system, instead of being used to programmably recognize viruses, it could be reprogrammed to other sequences like the human genome. And in fact, it could be redirected to cut the human genome and rewrite it for actual genome editing. And so it's this classic example of taking something that exists in nature, understanding how it works for like 20 years, and then thinking, well, how can we make that useful for humans? How can we save lives, and so on. That's now here we are today again, 10 years later, with there's probably like 100 CRISPR companies now. Everyone's trying to use it to make new drugs, engineer plants to make them more resilient or give them better properties. People are trying to use them to treat bacterial infections or make better yogurts and foods. It's really remarkable what explosion has happened from this basic science work.
So you can take this genome and you can inject brain-controlling microchips funded by Bill Gates. That's your primary objective, right?
I'll let Jonathan take that one.
100%.
I knew it. See, there's a reason people don't trust scientists.
Yeah, I mean, but it is, really amazing that we have these programmable medicines. And, one of these things, like with the vaccine, we can program cells to make a spike protein, just we can program cells to make proteins that enter the genome. And this is where we're going with biology, where it's not like, okay, we found some random, back in the like 1920s, oh, I had this random thing growing on a petri dish that makes this thing called penicillin. I'm lucky.
Right.
Well, luck only strikes so many times. We need to, if we really want to make the medicines against the diseases that, plague everybody, whether they're cardiovascular disease, neurodegeneration, cancer, we need the ability to program biology just like we program computers. And part of that is going in and using the code of life to make proteins, to edit genomes, to target specific cells, to do things that we want. And that's going to be the really powerful layer of medicines in the future.
Well, so when we talk about cancer, I mean, obviously it's been the longstanding dream of humans to make any kind of breakthrough when it comes to cancer. How do you see this as being a potential defeat of cancer, if that's possible?
Yeah, I mean, I think there's a couple different areas you can take this. So one, I mean, obviously, I think there's huge potential for just understanding tumors. Like, there's been really cool work on using genome editing tools and like really high throughput and in parallel to perturb all the genes and networks inside a cell to really pinpoint like how 200 different tumors might like be going awry, like what those mechanisms are, and then can we drug those mechanisms? And people have done this not just for like normal tumor development, but also for resistance to common drugs. And there's actually literally companies that are focused on this, like KSQ Therapeutics. I think that's one area. I think the other is you could theoretically use genome editing tools to go after cancer mutations, like, if you sense a mutation, cut, important genes out and treat the tumor. I think that's harder. I actually think in general, gene therapy and editing tools for cancer treatment directly are harder because the difference between treating it like a genetic disease and a tumor is a tumor you have to get every single cell. For genetic disease, right, like, you're missing a protein, maybe I only need like 1% correction, now you have enough of that protein to survive. But if I only got rid of 1% of your cancer cells, you would still have the cancer. If I got rid of 99.99% of your cancer cells, there's a good chance that cancer might come back. And so you have this problem where you really have to get it to every single cell, and that's hard. And that's not a problem necessarily with gene editing technologies. It's a problem with our current ability to deliver these technologies inside the body to different cell types. And that's called, those are delivery technologies, and it's a delivery problem that is not solved. So, just to give a little bit of perspective on this, part of the reason chemotherapies are so bad is you have like these really toxic compounds.
Right.
And to make sure you get every single cell, you're dosing them pretty high. And so they're going to hit like your normal cells. And the idea is you'll take that hit because the hope is you'll obliterate every single cancer cell. Right. And that's why those therapies, can kind of work. With gene editing, it's like, I don't think you want to dose a person so high that like you're getting permanent genetic modifications every cell in the body that could be really bad for you. The risk tolerance there is even higher, and even then you're probably not going to get every cancer cell. So that's why the approach has been more using these tools to understand the cancers and then making more traditional drugs based on those insights.
Omar, I know you've been working on aging and studying that for a very long time based on my research of you. And in researching aging and anti-aging and a lot of the new techniques, one thing that's come up over and over again is this idea of telomeres and telomerase and cell death and that kind of thing. Is that sort of related to stuff that you're working on? Is that a theory that you believe in, or have you heard of that?
Yeah, so I think there's many different axes of aging. So the telomeres, I think is one axis a lot of people have been working on. I think we're trying to take a more mechanism independent view. So, our approach is, can we understand actually how to reprogram cells to make them look younger? And part of that is like, can we study at the molecular level how young cells differ from old cells? That can be the proteins expressed, RNAs are expressed, how the genome is folded. And then can we actually figure out what are the exact right things to express to rejuvenate them? And there's been some work already done in this, like people have shown Yamanaka factors, which can make stem cells from like skin cells, right, can be used for actually reversing the clock on cells by expressing them at short periods of time. So you don't, make someone's skin turn into stem cells, but if you express them for a short period of time, the idea is that hopefully you revert them back enough that they look younger, and maybe that resets certain aspects of the cell to, make telomeres longer, or get rid of protein aggregation, or rejuvenate the mitochondria, right? But the idea is that if we can find more general ways to sort of reprogram a cell state, all the different factors of aging come along with that, not just focusing on the telomeres. So we've been doing a lot of that in our group, focusing on different tissues, trying to understand why maybe Yamanaka factors work on certain tissues but not others, and trying to find new ways of doing this. And I think it's really the early days of what people are calling partial reprogramming, but I think there's a lot of interesting promise in that direction.
So cool. I mean, it's mind-blowing, truthfully. So knowing what you know now, what do you think, from where you stand, is the most exciting thing that has the potential to be the biggest game change in the shortest, let's say, 10 years? What is the most exciting potential game-changing result from all of this?
Want to go first?
Yeah. It's tough. It's tough to say. I mean, I think that we're starting to really be able to develop medicines that can actually approach these areas that we've had good treatments, but not total treatments, that will have large impacts. Cancer is a big one. I think that now we'll be able to program cells and develop vaccines against cancer. So you see that, Moderna, Pfizer, BioNTech were able to make these programmable vaccines that we talked about for, COVID. And now that, we've kind of proven the concept there,
it's crazy.
These companies are now turning to things like, can we make personalized cancer vaccines?
And
so these technologies may usher in an additional era of new cancer treatments. There's ideas that we can use these for common diseases like cardiovascular disease. So there's a company actually in town called Verve applying genome editing to cardiovascular disease and can lower like people's lipids by editing their genome. So these things are, very sci-fi, but they're in clinical trials basically now. So I think in 10 years, we'll start to see the fruits of those where, genetic diseases are rare. You probably don't know someone with sickle cell or alpha-1 antitrypsin deficiency, to pick those names up, but you may. And rare disease day was literally yesterday. It's important to be aware of that. But there's also common diseases that may impact people directly. Yeah. It's also just more of a layer out of that, not just these programmable medicines, but just medicine in general, this concept of obesity management, and you may be familiar with the drugs Ozempic or semaglutide or Wegovy that are like burning through Hollywood because everyone's on them.
Right.
Those are actually incredibly impactful ways to manage appetite and diabetes that have had really like impactful like outcomes on people's weight, where they're losing 20-30% of their weight. So it's kind of crazy that people can just get an injection that helps them manage their weight. So we'll probably see more of those as well. One last one is pain, being able to develop new drugs for pain, non-opioid pain, where obviously the opioid epidemic was like terrible to this nation, but new drugs to be able to manage chronic pain, which is an outstanding issue, those are also in development. So we're, I think, going to see lots of very compelling new biotechnologies and medicines coming through in the next 10, 15 years.
I can't thank you both enough for sharing your time and wisdom, and obviously you're doing some incredible stuff. So if people want to follow along with your work, where can they find you? Where can they follow along with what you're up to?
Yeah, so we have a lab website, abugootlab.org. We have a lot of our publication and research on there. I would also say follow us on Twitter. We post really long tutorials about papers that we have come out, where we give sort of an inside scoop about the motivation and kind of the story behind the work, and try to give a high level view of what's going on every time a paper is published. You can also scroll through and find our old tutorials online about papers over the past many years too. So I'd say those are the best ways. Our emails are also online, so I also encourage anyone to feel free to reach out, and if you ever want to just talk about research or whatever, just we're available. So.
Well, that's fabulous. That's a great way to wrap it up. And here's hoping that you don't become the next Theranos. I'm trusting that you're both ethical. I'm just kidding, but no, it's been an absolute honor. Thanks again for sharing and for what you do and for what you're up to. And may all the random idiots in the world, such as myself, benefit greatly from your work in the next 50 years. That would be just incredible. And I look forward to following your work very closely. So again, thank you. And with that, the official podcast is over. Thanks again for listening to another episode of the Beat the Often Path podcast. If you've enjoyed this episode or any of the episodes we've shared, it would mean a great deal to me if you subscribe to the podcast on your platform of choice or on YouTube. And of course, if you shared either the show itself or this particular episode with somebody who might want to hear it to help us grow the audience for the show, I would greatly, greatly appreciate it. So if you've been a passive listener all this time, I get it. I understand. There's no big deal with that. But it would really, really mean a lot to me if you'd leave a positive review and help me grow this show. So thanks again for listening, and I'll see you next time.